is a graduate student in biology, obviously, but she graduated from JSU with her Bachelor's of Science in Biology in December 2010 and has been working diligently for the last 2 years amidst teaching labs as a graduate TA. In addition to, she also worked in the private world for about 8 months in the midst of all of her thesis, and yet she still was able to conduct a very extensive lab and field study on some fairly difficult questions, all within a 2-year time period. So she's definitely an example of success for our department. And with that, I'm going to just put that on as your no pressure. I would like to introduce the title of Megan's thesis project, which is Biological Integrity of Snow Creek, a Historically Impacted Stream.
More specifically, we looked at an index of biotic integrity. And what this is a measurement that tells us or allows us to determine the quality of a stream based on the presence or absence of specific indicator species. And it actually gives us a numerical value that we can compare between different sites. So this is an overview of all of the study sites used throughout the study. If you see over on the right, you can see this is the Talladega National Forest, where one of our sites is located. This is the Jacksonville area, and this is the Oxford area. And then you can see over here, this is the Coosa River drainage. It's important to note that the areas highlighted in yellow are heavily urbanized, or they are urban areas. So again, the first thing we looked at was metabolism. And we wanted to look at 3 different sites within this study. We chose, our first site was Shoal Creek, which is located in the Talladega National Forest. And we chose this site because it's a relatively pristine site, meaning it does not have a lot of anthropogenic influence that is affecting it. This is our reference site for the study. And the next site we chose was Mill Creek, which is a slightly urban site. It's located here in Jacksonville. It is urban, but we know that there's no indication of PCB or mercury contamination at that site. And then, of course, our last site is Snow Creek in the Anniston-Oxford area, where we know there is both urban and PCB and mercury contamination. So the main thing with this is we chose these sites because we wanted to try to tease out the effects that the mercury and PCBs were having on the fish there. So that's why we chose Mill Creek as our urban site and Snow Creek as our urban plus contamination site. Because you would assume that if you saw a change or a difference between the physiology of fish from Snow Creek in comparison to fish from Mill Creek, then you could potentially assume that there's something other than urbanization affecting the fishes that are living there. So we collected fish using backpack electroshocking techniques. And the model organism that we chose to look at was the large-scale stone roller, Capistoma oligolepis. We chose this guy because he is a very ubiquitous shiner species. He was consistently found at all 3 sites, and we needed that consistency to allow us to conduct the study and have enough samples to run our analyses. So again, this is the study area. You can see Highway 21 again right here and Interstate 20 running east to west. So we had our Mill Creek site in Jacksonville, our Shoal Creek site in the Talladega National Forest, and Snow Creek in Oxford. So this is a picture of Shoal Creek, and this is also to prove that I actually did get out in the field. This is our Shoal Creek site in the National Forest. You can tell that this looks like a beautiful site. It has great aquatic vegetation. The riparian zone is gently sloping, and there is a lot of canopy cover. All of these things are indicators of a good quality stream or a quality habitat. And this is our Mill Creek site in Jacksonville. You can see that there's still riparian vegetation but there are some steeper banks and it is slightly impacted. But this is Snow Creek. And I don't know if I need to say too much about this. It's pretty obvious that there's a train track on one side and on the other side the bank is concrete. That's obviously pretty indicative of limited habitat and a very stressed system. So again, the first thing we looked at was respirometry, and what we wanted to do was examine basal metabolic rates using closed system or closed chambered respirometry techniques. So we looked at basal metabolism, and that is just the resting metabolism of an organism. So it is what the most basic metabolic needs an organism has to maintain normal physiological functions. So fish were collected in the field using backpack electroshocking. They were transported back to the lab and separated into 3 tanks based on their site of collection. Over a 2-week period, the tanks were brought to a holding temperature of 20 degrees Celsius. So slowly we changed the temperatures in the tank depending on their site of capture. So if the stream that we collected the fish from were 15 degrees C, the tanks were held at 15 degrees C and then slowly brought up to 20 degrees so that we would not elicit any changes in metabolism due to stress from the tank systems. They were also on regulated light cycles. This is also of concern when you're dealing with metabolism. You don't want to change any of their circadian rhythms. So we placed the tanks on a 12-hour on and 12-hour off light cycle. Feeding was also restricted because, of course, if you eat something, your metabolism is going to change. So 48 hours prior to any measurements, the fish were not fed. And then before being placed into the respirometry system, or the respirometry chamber, which is what you see at the bottom, the weight of each individual was measured and recorded. And you can see that's the stone roller in his respirometry chamber. So, if you look at the top diagram, you see an overview of the entire respirometry, or of the respirometry system. Again, the fish would be placed in the respirometry chamber, or the fish chamber here. And the way the system works is you have a flush cycle and a recirculating cycle. And so the first thing you're going to go into is your flush cycle, where the organism is going to be, water is going to pass over the fish and throughout the system, and it's going to be fully oxygenated. After about a 2-minute lapse, the flush cycle will stop and the recirculating cycle will start. And that's when you're actually going to take your measurement. So the same water is going to pass across the organism for a certain amount of time. And what happens is the fish will consume oxygen from the water, and an oxygen sensor up here will read how much oxygen is being removed from the water. And it gives us a graph so that we can see the decline in oxygen. So to make this more relatable, if you were to go to the doctor and they were to run a stress test on you, it's the same thing. They put you on a treadmill, hook you up with a mask, and measure the amount of oxygen consumed and carbon dioxide released. Same concept. We put our fish on a little treadmill, just so happens it's just a fish chamber that water flows past, and he consumes oxygen over a certain amount of time. So after the recirculating cycle ends, the flush cycle will start again, and the process will be continued at least 4 times. We do it 4 times so that we can make sure that the fish have a chance to become acclimated to their environment, and they're not showing changes in metabolism due to handling stress or being in a confined space that they're not used to. So at the bottom you can see our 3 holding tanks. Where the fish were separated based on site. And then on the right you see the entire respirometry setup. It's also important to note that the respirometry system and all of the water was also held at 20 degrees C because we want to make sure that temperatures are the same between the tanks that they were in as well as the respirometry chamber. You see the computer to the left. We have a analyzing software that measures the amount of oxygen that the fish consumes, and it converts it into a metabolic rate. So we all know that fish are aerobic organisms, and oxygen consumption is directly indicative of metabolic rates. So we are given a metabolic measurement known as an MO2, and that is milligrams of O2 per kilo hour. So what this measurement is telling us is the amount of oxygen an organism is consuming per weight per amount of time. So that's why when I said earlier we measured each organism before we put him in the respirometry chamber, his specific, or the individual's specific weight was factored into each calculation. This is important because it allows us to compare individuals of different weights. So you can see here our first bar is Snow Creek, Mill Creek, and then Shoal Creek. The MO2s, or the metabolic rates, ranged from approximately 200 to 250 MO2. And there was no statistical difference between the oxygen consumption or the metabolic rates between any of the sites. Just to be safe, we chose a weight class that ranged from 6 to 12 grams for each of our fish, or for the groups of fish that we ran. And to make sure that there couldn't be, or that there weren't any variations in the weight classes that we chose, we also did a statistical analysis to look at the weight class for each site. And again, there was no statistical difference between those weight classes. Okay, so the next thing we decided to look at was gill morphology. And we did this using scanning electron microscopy techniques. Fish were collected from 2 different sites. We chose Snow Creek and Mill Creek. We wanted to see if there were differences, of course, in the gill morphologies from these 2 sites. So this is the procedure that we followed to fix the gills of these organisms. What we did was the fish were collected in the field. They were immediately brought back to the lab and held in the same water that they were collected in overnight. The next morning, they were euthanized, and gill arches were immediately removed from the fish. At that point, they were placed in a 0.9% isotonic solution, and they were washed 4 times. The next step was the primary fixation in 3% glutaraldehyde, and we repeated this process twice with 1-hour washes, or 1-hour fixes. And then the secondary fixation was in 2% osmium tetroxide, and we again did that twice at 1-hour washes. And then we did a serial alcohol dehydration step. And at that point, we wanted to make sure that we had removed all of the water from the gills, or from the tissue. We did this at 10% increases for 1 hour each. So we started at 50% isopropyl alcohol, and it stayed there, or it washed for 1 hour at that step. And then we would move on to the 60%, 70%, and sequentially until we made it to 100% isopropyl alcohol. And then the last step was a drying step, and we dried our gills using hexamethyl disilazane. We did this for 2 5-minute washes. After that step was completed, we let the gills fully dry under a ventilated hood before they were placed on a SEM stub with carbon tape and then sputter coated with gold palladium. So to give you an indication or an idea of what gills would look like if they were damaged, if you look at the top 2 pictures, you'll see gills that were damaged by heavy metals, and the bottom 2 images are gills that are damaged by PCBs. So some of the typical indicators, and this is pretty inclusive for all of both PCBs and mercury, is you'll tend to see swelling of your gill lamellae, and I guess I should tell you a little bit about the gill anatomy first. So each of the gills have a gill arch, which is what you would see right here and right here. And then you have gill filaments, and those are these filaments coming off. So each of these are a filament. And then above and below each filament you have lamellae, and that's the actual site of gas exchange. So that's where the water would run over the lamellae and oxygen would be absorbed or consumed by the organism. So to go back, one of the major indicators is lamellar swelling. So you can see these are the lamellae, and that is an example of lamellar swelling. And then if you look here, you'll see lamellar fusion, and that's where the lamellae have actually swollen to a point that it closes the gap that the water would typically run past. So it limits the amount of oxygen exchange that is even capable of an organism. A lot of times, the gill swelling is often attributed to an immune response, so it's an inflammatory response. And one might think that since this is such thin epithelial layers, that remove them or reduce the amount of pollution or toxicant and the lamellae or the epithelium will replace itself. But another issue is that with these mercury and PCBs, the lamellae or the epithelial layer tends to become very thick, and it doesn't replace as easily as it would if it were exposed to some other pollutants. Okay, and then at the bottom, you can see again, this picture is actually using a light microscope, so it's a little bit different, but you can see that these are the lamellae, and there you can still see the swelling of the lamellar fusion. There's no space between the lamellae. So this is these are images or micrographs of the gills that we actually examined. So if you look at the top left, that is the those are the gill filaments from a stone roller from Mill Creek at 150x magnification. So at least 5 individuals were examined from each site for this study. If you look at the right, again, that's 150x for Snow Creek. And so you can tell that there's no difference, or there's not a large difference between the gap between the lamellae of the fish from Mill Creek versus Snow Creek. And in addition, you can tell that they are no way as swollen or as close together as the lamellae of examples of damaged gills. So again, at the bottom you see Mill Creek at 1500x and Snow Creek at 1500x in figure D. So again, we did not see any indication of lamellar swelling or fusion in our fish from Mill or Snow Creeks. So the last thing we looked at was fish assemblages. To do this, we had to collect fish, and the method of collecting fish followed the EPA rapid bioassessment protocols. We did this through backpack electroshocking and seining techniques. So if we were in an area where we knew there were sensitive taxa or sensitive species located, we would not use an electroshocker. We would typically use seining techniques. And this is also, this technique is also called green sampling, because what we do is we'll go out and we'll collect fish, we'll identify them on site and actually release them at the site of capture so that we're not sacrificing any of the individuals. So we had 8 collection sites for this portion of the study. Shoal Creek at Pine Glen, which is in the Talladega National Forest. Eganaiga Creek at the confluence of Choccolocca Creek. Choccolocca Creek at Cider Ridge Golf Course. Hillaby Creek, Snow Creek at an upstream site, so closer towards Anniston, moving north. And then Snow Creek at the confluence with Choccolocca Creek. Coldwater Creek and Choccolocca Creek at Preeb's Mill. So this is a map of all of the sites that we looked at or we surveyed. You can see Talladega is more isolated. Again, Shoal Creek is more isolated. It's in the National Forest and receives little impact or little anthropogenic influence. Site 2, Eganaiga Creek, again, it's not necessarily considered an urban site. It is slightly impacted by agriculture, or it is impacted by agriculture. And you have Hillaby Creek and Choccolocca Creek at Cider Ridge. And you can see here, this is the base of Cheehaw, sorry. Again, you can see Anniston is obviously a very urbanized area, and this is or Oxford, Anniston and Oxford. This is our Snow Creek site upstream and our Snow Creek site at the confluence. And then we have Chockalacca Creek at Preeb's Mill, and then we have Coldwater Creek. So this is just a closer image. Shoal Creek is not in this map, but this gives you a better idea of where the other sites are located in relation to one another. So we collected fish at all of these sites, and a total of 1,087 individual fishes were collected in total. 39 species of fish were collected from all 8 sites. And the top most abundant species were the stone roller, which you can see right here, the bluegill sunfish, the Alabama shiner, the tricolor shiner, and the banded sculpin. So these top 5 species made up almost 50% of the population of fishes we collected in total. This is a chart showing all of the species that we collected, the number of individuals we collected, and for each site, rather. I wanted to point out something on this chart because I know it's just a bunch of numbers and names, but we know that Snow Creek has been historically impacted, and everybody thinks of it as such a impacted site. But if you look here at Site 5 and 6 are our Snow Creek sites, and if you look down at the bottom, we typically think of darter species as being more sensitive. And what we're seeing in Snow Creek is that there are actually some darter species that are showing up there. So that could be a good indication, but that's something that would need to be looked into further. So the next thing we did was looked at species richness. So what was the total number of species that were collected at each of the sites? These results actually grouped into 3. So our first 2 sites being Shoal Creek and Eganiga Creek had a total of 19 species at each site. Then sites 3, 5, 6, and 7 fell in the middle with 15 species. And sites 4 and 8 had a total of 11 species. So to take a look at this a little bit closer, we looked at species diversity using the Shannon Index. This not only takes into consideration the number of species there, but also the abundance of those species. So it gives you a little bit more detailed explanation of what is actually occurring in these populations or at the community level. So we found that Shoal Creek had a Shannon Index of 2.09. These indices typically range from 1 to 5. A study conducted within the region indicated that Shannon indices for this general area range from approximately 1 to 3.5. So based on that, our Shannon indices fell within range. But none of these were really out of character. Shoal Creek did not have the highest Shannon Index, but the survey that we were looking at was heavily populated with 1 species. So due to that abundance of that 1 species, it kind of outweighed some of the other sites. And so the last thing we really looked at was our index of biotic integrity. Like I said earlier, this is a quantitative measurement that tells us the quality of a stream based on specific species. So this list on the left, there are 12 metrics that are used to calculate an IBI. This has been recently updated for the actual ridge and valley region of northeast Alabama. So based on our IBI scores, we found that Shoal Creek had the highest IBI rating. 6 of the 8 total sites had an IBI rating of poor to very poor. And so again, Shoal Creek had a good IBI reading, as well as Eganiga Creek, which had a fair reading, and the rest of the sites all indicated a poor to very poor. So again, this is just an easier way of looking at this. This is the scale that is developed for the IBI for the Ridge and Valley region. So what is this telling us? We know that Snow Creek is poor to very poor, but it's also surrounded by a lot of other streams that are poor to very poor. So we can't say that Snow Creek is more or less impacted, or in this case, we can't say that it's more or less impacted by the PCBs in Mercury, because it is falling in line with other streams in the region. I think what it does tell you is that all of the streams within this watershed, or within that region of the watershed, have been impacted negatively in some way from either urban or agricultural practices. And this is a picture of a speckled darter, which is one of the species that we actually find in Snow Creek. So in summary, based on our physiology and anatomy indices, or the things that we were looking at, fish of Snow Creek show no abnormalities or no additional stress in comparison to other streams within the watershed. So like I said earlier, this does not mean that Snow Creek is great. I think what it's really telling us is that all of these streams are impacted in some way. And another thing to take into account with this is that although we did not get results with the stone roller that we were looking at for gill malformations or alterations in metabolism, it could be noted that a more sensitive taxa might indicate changes in both gill and metabolic rates. I say this because stone rollers are a very tolerant species, or they're not necessarily listed as tolerant, but they're a very hardy species. And so because of that, we chose the stone roller because we knew it would be found at all three sites. So there's a caveat to this. It's difficult to use a sensitive species because you can't necessarily find the abundance that you need to run your statistics or to run your study. So it may be useful to try to find a sister taxa that is a little bit more sensitive, that would potentially indicate stress at the gill or metabolic level. And then, in addition, there are more sensitive techniques available. The use of CYP1A has been shown to alter in response to, or to show up in response to, chronic exposure to both mercury and PCBs. So more sensitive techniques could also be used to indicate this. And then again, the last thing we looked at was fish assemblages. And based on current IBI scores, the condition of Snow Creek is similar to other streams within the watershed. I think this is probably the most indicative evaluation that we did for the system itself for Snow Creek and those surrounding streams. They are impacted, and they are showing some type of influence from this urban and agricultural environment. And with that, I'd like to thank everybody. So they'll bed down in the sediment, and they're also long-term persistent pollutants, so that they will remain in a system for decades and slowly release into the water column if sediment is stirred up or moved. So yes, the organisms do continuously come into contact with the mercury and PCBs that remain. Did that answer your question? Yes, sir.
We know what Snow Creek being affected by, but also you said the urban environment influenced other streams. What do you think? What type of impact it comes from where and what it is?
For urban, we have a lot of industrialization, so runoff. The fact that the creek runs underneath the mall and Interstate 20 is, I think, a big impact there. And then there's also a lot of storm water permits that are still being given to industries in the Anniston and Oxford area, which allows them to release runoff directly into the stream if there is high rains.
In the last one, with the abundance of the species, why didn't you go to the site you showed, the Christine site, and use that as a background?
I'm sorry, I don't understand.
The first slide when you were in that beautiful scenery, which is the Christine. Have you used that as a background for the abundance of the species?
I don't understand what.
Yeah, you use Shoal Creek as
yes.
one of your notes.
Okay, I thought you meant how did I use it. No, I did use, yes. Shoal Creek was my...
You seem is impacted too?
Do I think Shoal Creek is impacted? Yeah, although it is, in a protected area, it's still impacted by forestry and prescribed burns. So there are still some impacts that are taking place. It's just not to the extent of what's going on in the Anniston-Oxford, Lower Choccolocca, Middle Choccolocca area.
It was your good side on the IBI, wasn't it?
Mm-hmm.
Okay.
It
It was
was.
the only good one she had on the whole
Mhm.
slide.
Yeah, it had an IBI rating of 48, which is very good.
50 is
yeah.
nice.
50 is very good, actually.
What about Stone Creek?
Snow Creek, one side on Snow Creek fell in the poor range, and the other side fell in the very poor range.
The north or the south?
The northern side was the very poor, and the side at the confluence was just poor.
Can I make a comment?
Yes.
Actually, two comments. The very first slide, actually, can you show very first slide or maybe second slide? There is, basically you are saying that it is okay to have 1 microgram of mercury per gram,
Oh.
right? Oh, yes, this was
This
this one. 1.41 micrograms per gram.
Mhm.
This is quite a lot. Is this recommended?
Yeah.
One ppm?
In Europe, a lot of countries have lower limits, but in the US, the FDA permits
1
fish to
ppm.
be sold that has that.
Yeah, EPA,
concentration.
So that's
Yeah, 1
really
microgram
very
per
high,
gram.
very high.
The EPA, they list that as acceptable range.
Mhm.
But worldwide, a
It
lot
is
of
much,
places that's
much lower.
considered
The high, much more
high,
high.
yes.
Well, it's probably.
Second comment,
Mhm.
I see that you are using osmium tetraoxide.
Mhm.
Folks, you should be extremely careful with this compound. This is extremely toxic compound. Extremely
It'll
toxic.
fix your
Put it in our
irises in your eyes.
Especially young people.
Retina sign.
Like you are, basically, because this is a compound that could have very, very negative impact on.
How much lecture did I give you before I let you even look at it? So,
Yeah,
That's
I was
insane.
told exactly how to handle it. I
Ah.
wore gloves. I even wore like big mitts when I
Yeah, I
opened this
understand,
thing.
but you see, the problem is that this is very volatile compound.
It is,
It
mhm.
smells like garlic, okay? And
she was under a hood. We used
so
proper safety
very
techniques.
careful, very careful. This compound is very, very dangerous.
Well, I've seen some Mad Hatter type behaviors coming out of you. Maybe
It's the osmium tetroxide.
I thought it was the mercury from walking into the
Yeah.
water.
Maybe just to satisfy myself whether you learned from my class. Now, you have a 3 site and you analyze for the metabolic rate. To compare the 3 site, what type of statistical analysis you use?
Used in ANOVA.
Why?
Because we wanted to compare the statistical difference between the means of the 3 groups.
And what F value show? There is
I don't know.
no significant difference?
Yeah, there was no significant difference. And so because of that, we didn't do any further
And
analysis.
what type of ANOVA you use? You block it or you CRD or block?
Block.
Very good.
Taking me back a couple of years. Yes, sir.
I know very little about biology, but I'm just wondering, why did you study gills and not say liver? If mercury is damaging something, it should rather damage liver. Am I correct on this?
Oh, sorry. Actually, but mercury and PCBs, of course, they're both lipophilic, and they do accumulate in the liver of organisms. But in addition to that, they also accumulate in the gills, because the gills are the primary site of contact with an aquatic pollutant.
So it was the first contact. Right?
Yeah, it is. So that's why I looked at the gills for morphology.
They're really sensitive to
They're
anything.
very, very sensitive to, Jenny.
If you were looking at CYP1A, what tissue were you going to use?
Liver. But CYP1A is also elicited in the gills and intestines of these fish as well.
It's P450 class of enzymes that
Oh.
is expressed greater with mercury.
Yeah, metabolizes pollutant.
Anything else?